Alright, protein synthesis. First off, you have DNA. DNA consists of a double helix, is made up of 20 amino acids in a long polypeptide chain, has 4 bases, adenine, thymine, cytosine, and guanine, and then has the base pair rule, which states that adenine and thymine will always bond together, and cytosine and guanine will always bond together, which you can see in this photo right here. There's 2 types of bases. There's pyrimidines, which are single ring, and purines, which are double rings, which you can see right here. You have scientists involved are Rosalind Franklin and Maurice Wilkins. They took diffraction video or X-ray photos of DNA crystals and then gave these photos to Watson and Crick, which then they used the photos to build the first DNA model, which you can see right here, and basically took all the credit for it. You have DNA replication. DNA replication is basically you're going to be copying the DNA to make more sets of the DNA. Right here, which is when the DNA chromatin is copied to create more DNA strands. Each strand has an original copy, which is semi-conservative. The process consists of the helicase enzyme unwinds the DNA by separating the hydrogen bonds, which is the weak bond in the middle, and from the nitrogen bases, and then DNA polymerase, another enzyme, bonds these nucleotides into strands and then proofreads the new strands to make sure everything's in order, and then the DNA is rewinded back into its original state using DNA ligase. This is a photo example, DNA replication. It's also another example. This is this would be the first original strand, and this would be the copied strand right here. Mutations. Mutations are a change in DNA sequence and is only passed down through gametes, which are sex cells, and non-somatic cells, which is basically just normal cells in your body. There's different types of mutations. You have chromosome mutation, which is a change in the sequence of a chromosome, and these have a larger impact, not necessarily more harmful. Some of them are harmful, some of them are not. And then there's gene mutations, which are a change in the structure of a gene, and these tend to have less of an impact on everything. There's different types of mutation. You have deletion, when a whole section of the chromosome is deleted. You have inversion, when a part of the chromosome is flipped. You have translocation, when 2 parts of a chromosome switch spots, and non-destruction, when the chromosomes don't separate during meiosis. And then duplication, when a gene sequence is repeated. Then there's gene mutations, 5 types of gene mutations. You really only need to know 2, which is frame shift and point mutation, which is when you insert or delete more than 1 nucleotide, and then point mutations are when there's a change in only a single nucleotide. Frame shifts tend to have more impact on the gene. DNA and RNA. DNA is double strand and contains a ribose sugar, while RNA is a single strand and has deoxyribose sugar, and it has U's instead of T's for one of its bases. Alright, types of RNA. There's the messenger RNA, which copies it and carries genetic info into the ribosomes. There's ribosomal RNA, which basically makes up the ribosomes, and then there's transferred RNA or tRNA, which transfers amino acids to the ribosomes. Alright, right here you have transcription. Transcription is when DNA is copied and moved out of the nucleus to a ribosome. This whole process uses mRNA or the messenger RNA to copy the original DNA, and then the RNA polymerase is used, which is an enzyme, is used to split the strand and make the copied RNA. And then you have translation. Translation involves transferring the DNA into a protein, so this whole process is just transferring it from the nucleus into a protein or amino acid. This uses tRNA to read the mRNA, and this is all happening inside of the ribosome. This takes the tRNA and it reads the mRNA and brings it to the correct amino acid. What it's bringing into the amino acids are codons, which are groupings of 3 bases. So like you have A, T, C, or for example, and then you have anticodons, which is part of the tRNA that reads the mRNA codon and brings it to the correct amino acid to form the protein. Amino acids it has 20 different types, can contain multiple different codons, So some codons tell the ribosome to stop translating, which you'll see in a bit. So you have the steps. You have the initiation, which basically the start codon, it tells it to start translating. You have, I don't know how to say that, but you have empty tRNA is kicked off, peptide bond is formed between amino acids, and new codon is brought in. And then you have the termination, which is when the stop codon stops translating. Alright, and that's basically it.